Good morning and welcome to the Emerald Ash Borer Biological Control Release and Recovery Guidelines Workshop, Module 1. So the goal for this workshop, the USDA remains committed to managing EAB, and it has prioritized management through biological control. So we want to give you the tools and resources to be able to confidently choose an EAB parasitoid release site, conduct your releases, and conduct your recoveries. In addition, this is a nice opportunity to connect you with people involved in the EAB Release and Recovery Program so that you can feel free to contact us in the future if you have questions. This is also here to help you understand in general the guidelines and also the science behind how we develop those guidelines and why some of our rules and guidelines are the way they are. All right, so I just wanted to give you some photos of me and kind of put an image to the person who's speaking for a lot of these modules. But my name is Theresa Murphy, and I've been working on biocontrol of insect pests since 2011. And I've been working on EAB biocontrol since 2013. I received my BS in Environmental Science and Management from the University of Rhode Island and my master's in Organismic and Evolutionary Biology from the University of Massachusetts Amherst. My master's research focused on factors influencing the impact of biocontrol agents of the emerald ash borer. I've been with the USDA APHIS PPQ S&T at the Forest Pest Management Lab in Buzzards Bay, Massachusetts since 2017. I focus on the biocontrol of emerald ash borer and Asian longhorn beetle in both the field and the laboratory, but I've also worked with spotted lanternfly and Polyphagous shothole borer. And here are some photos showing some of the really fun things I've seen while conducting EAB releases and recoveries. So I saw the 17-year cicadas emerge out in New York while doing some parasitoid releases. I've also seen frogs, and while doing recoveries, I saw a juvenile woodpecker on one of our recovery trees, which was pretty fun to see. This is from my master's research where I caged in parasitoids so I could look at the difference of how woodpeckers and parasitoids interact. So in Module 1, we'll go into an introduction and background in life cycle of EAB. Then we'll talk about how to identify EAB and possible lookalikes, as well as going into the history of biocontrol of EAB. Then we'll look at how to identify ash and possible lookalikes. The next session will be impacts of the EAB parasitoids with Dr. Julie Gould, and then a quiz section on tree species and insect ID. So we're going to start by going through the background of EAB. So many of you are probably very familiar with the background of EAB, so I'm just going to cover it briefly here. Emerald ash borer is an invasive beetle originating from China that was first found in the Detroit area of Michigan in 2002, although later studies have determined that it was likely introduced into the United States in the early 1990s. And as of March 2021, it has been found in 35 states, the District of Columbia, and 5 Canadian provinces. And below you'll see a little timeline showing the different states' first detections over the years. So I'm just going to show you the map of the spread as it has spread since I started giving this presentation. So we first did this presentation in October 2019. You can see the map there. It spread more in 2020 and then 2021. And since then, we've actually discontinued that federal quarantine. So the latest map shows the spread with just the new county detections highlighted in green and purple there, and all the existing county detections in this light blue color. The majority of our North American fraxinus species are known to be susceptible to the emerald ash borer, including white, green, black, blue, and pumpkin ash. Up to 2% of the country's total leaf area is estimated to be ash, and ash is a vital component of many ecosystems. It's used for a variety of applications, including basketry, tool handles, baseball bats, furniture, cabinetry, solid wood packing material, pulp, and paper. And basketry has cultural significance as well as that social economic significance. It is estimated to cost tens of millions of dollars per year to manage emerald ash borer, and it causes approximately $1 billion in damages per year, and it threatens resources worth an estimated $300 billion, including an annual timber value of about $25 billion. Originally, it was under quarantine and eradication. Now the federal priority is on management through biological control. So how does EAB kill a tree? The larvae themselves feed on the phloem cambium and outer sapwood of the tree. The galleries from this feeding cut off the flow of nutrients, which stresses and eventually kills the entire tree. Emerald ash borer generally initiates its attack in the canopy of the tree, and trees with only a few galleries may show no outward signs of damage, making diagnosis very difficult in the beginning of an infestation. So here's a nice graphic that shows, again, where that feeding is occurring, right in that cambium tissue layer of the tree, which cuts off the tree's vascular tissues, which disrupts water, mineral, and nutrient transportation throughout the tree. EAB is having significant forest impacts, especially in areas where ash is a primary component of the ecosystem. You can see an example of that in the photo on the left. And ash has a wide distribution in the U.S. There are over 150 insect species that have been recorded feeding on ash leaves and several dozen insect species that rely solely on ash. Birds and other wildlife will feed on those insects as a food source, and ash seeds are also an important food source for a variety of wildlife. An open forest canopy caused by dying ash can have cascading ecological effects. It can promote the invasion of non-native plants, decrease soil moisture, and threaten nearby tree species. This photo shows an invasion of multiflora rose in an area where we were working in New York, and we worked there for several years so we could see as the ash started to die and the canopy opened up, this multiflora rose spread to many areas to the point where we basically had to swim through it to get to our trees to do our ash health assessments. EAB also has a significant effect when it kills street trees in urban and suburban areas, where ash is one of the few green spaces in those areas. Mature ash trees provide important shade, and especially in cities, they can reduce urban heat island effects. However, less noticeable may be that urban green spaces can promote mental and physical health. This article that I show here found that the loss of trees to the emerald ash borer was linked to an increase in mortality related to cardiovascular and lower respiratory tract illness. And it's just one finding adding to a growing body of evidence that urban greenery promotes major mental and physical public health benefits. Next, I will go through the life cycle of EAB as well as signs of an EAB infestation. Adult EAB typically start emerging around 400 to 500 growing degree days and peak around 1,000 growing degree days. For reference, in New England, that means they start emerging in May and peak in June. Adults are strong flyers. They feed on ash leaves, and they start to mate after 1 week. They begin laying their eggs 2 to 3 weeks later. They mate and lay eggs during the afternoon, and one female can lay over 200 eggs. These eggs are white when they're laid, and they start to mature and turn red after a few days. The eggs will typically hatch within 2 to 3 weeks of being laid. Then the larva will start to feed. The larva will continue feeding until the weather gets too cold. The mature larva will, if they are ready, overwinter in pupal chambers, and some larvae may require another season of feeding before they can complete development and become adults. Once they are ready to become adults, they will pupate and then emerge that following spring. EAB takes 1 to 2 years to complete its life cycle, depending on temperature, latitude, altitude, and the density of EAB in the tree, as well as the health of that tree. The map here, which I will talk about again when we talk about where it's appropriate to release different parasitoids, but I just wanted to point out the map now because it's very useful in showing where we kind of expect EAB to be in a 1 or 2 year life cycle. So we monitored emerald ash borer overwintering stages at 90 sites across 22 states with the help of many, many collaborators, and then modeled that and linked that to the growing degree days at those sites to help predict where you would expect your 1 and 2 year life cycles for EAB. So this shows the predicted probability of EAB overwintering as larva, which is the probability of EAB in a 2 year life cycle. And it shows throughout most of the US, there's a mixture of 1 to 2 year life cycles expected. And then in the South, majority 1 year life cycle. And in the very northern areas of the United States, a majority 2 year life cycle. Now we'll start to look at some of the signs of EAB damage that you can see when investigating whether trees in your area are infested with emerald ash borer. So one of the common signs that people start to look for is bark splits, which is caused by callousing around the old galleries of the tree, as well as those very distinctive D-shaped exit holes. So here you can see some more callousing around a tree that shows a gallery. Usually you don't get something so distinct, but oftentimes here you'll see another bark split, which is caused by an old gallery underneath, as well as another one of those D-shaped exit holes. Another sign that we look for is yellow foliage, dying branches, dead branches, small leaves on those branches, even in the full height of summer, and then thinning crowns. The next tool I want to talk about for assessing EAB damage is crown class health assessments. Now, looking at crown classes is used for a variety of tree species, but with EAB in particular, Smith in 2006 developed this scale that has been used by a lot of researchers since, which is a 1 to 5 scale for damage in crown classes for EAB. On this 1 to 5 scale, a 1 is a completely healthy tree that shows no signs of EAB infestation, no dieback, no thinning crown. Now, if you have an understory tree, you may see that the leaves aren't as full as an overstory tree that gets full sunlight, but the important thing here is that it looks completely healthy with no signs of damage. A 2 is a tree that is starting to show some signs of damage, but is still fairly healthy. There may be crown thinning, but there's not a lot of dieback at this point. There's still a lot of leaves on the tree. A 3 is a tree that's starting to show some significant signs of damage. Dieback is happening, leaves are being thinned out, but there's still a lot of leaves on that tree. A 4 is a tree that is showing the majority of the branches and the leaves are starting to show significant signs of dieback. There are still some leaves that's still alive, but in this photo you can see the crowns, the tops of the crowns are almost completely dead. These trees are showing significant signs of EAB damage if they're in a 4 crown class, and then a 5 is trees that are completely dead. And this crown class assessment, this guide of quickly identifying trees on a 1 to 5 crown class scale is very useful for when you're looking at sites and trying to assess if a site is suitable for releases, because you're looking for sites with mostly healthy trees with only a few trees that are in 3 or 4 crown classes. And so this scale is useful to know for doing site assessments. Another sign that I want to talk about for EAB damage is epicormic shoots in basal sprouts. Basal sprouts are basically epicormic shoots that are coming out of the base of the tree or sometimes out of the root system. And these signs, these shoots, are basically a sign that the tree is stressed and is trying to get more resources. They are caused for other reasons, but they're very common in EAB infestations and are a good indication that there is a stressed tree. So the way to tell these shoots from branches is that they're coming out of the trunk of the tree. A lot of times they're very thin looking, and they come out and then almost go straight vertically, as opposed to branches, which usually come out further than that. You may also see epicormic shoots in the winter or older epicormic shoots that have lost their leaves. So when they're dead, they look just like these thin branches, like I said, coming directly out of the trunk. going in most cases almost vertically. And again, is a sign of stress. And we'll often talk about if a tree shows more signs of stress, it will have a lot of epicormic shoots that will be present throughout the crown and the base of the tree. If it is a less stressed tree, you may just see a few epicormic shoots on one section of the tree. Another sign of EAB damage, which I'll say is probably the biggest indicator that's used to find new infestations because it's such a visible characteristic to look for, and it shows that there's definitely a beetle infestation within a tree, is woodpecker feeding. So woodpecker feeding can take a variety of different forms as the woodpeckers start to flake off bark, but woodpecker holes, woodpecker feeding that we look for, particular with EAB, is looking for a clear indication that a woodpecker has gone into the tree and taken something out, in this case a larva. So if you're looking, you may see flecking, bark that's been scraped off, and this can happen even when a branch falls on a tree or something like that. But if a larva has been taken out, you'll see this deeper hole that goes into the tree, and that's an indication that the woodpecker is eating larvae out of the tree. There are some native beetles that we'll talk about later that woodpeckers could also be feeding on, but very often, if you're in an area where there's EAB infestations, this means that the tree has EAB. And here you can see in this one, woodpecker feeding that is older. So when it's fresh, it's much lighter color than the bark, but as it gets older, this is probably at least a year or so old, that wood starts to fade again, and so all you see is the hole that's going in. The flecking on the bark is not present anymore. So these are two holes that are older for woodpecker feeding, and these are holes that are fresh. Another term that you might hear is blonding on a tree, and blonding is just when the woodpecker is flaking off bark, and a lot of trees that are stressed and filled with a lot of EAB will end up with flaking over large sections of the tree. So in this case, there's flaking off of the bark all over, and this is from woodpecker foraging. Although the actual holes of where they went in are probably just a couple, it's very visible to see the flaking on the majority of the tree, and we call this the woodpecker blonding. There are different signs as an infestation progresses, and it can be very hard to see sick trees right in the beginning. So trees with only a few galleries often show no outward signs of EAB damage, which can make diagnosis really difficult. By the time the tree is showing a lot of signs of stress, it usually has multiple galleries within it. And an EAB generally initiates attack in the canopy, which again makes it harder to see those signs of infestation because in mature trees, the canopy is very high up. So you're looking for exit holes and bark splits that are way up in the top of the tree that you wouldn't see without binoculars. So in the beginning, you can expect a few dead branches or maybe just some thinning leaves, a few epicormics, but it can be very hard to see an infestation until it progresses. When it progresses, you're more likely to see that woodpecker damage, severe dieback, damage in the lower part of the tree that's easier to see at eye level. And this is more common a few years into an established infestation within that tree. And so because it continues to be difficult to detect EAB at low levels, once it's detected, communities often experience a rapid loss of their ash trees. This figure here shows the years after an EAB infestation along with the percentage of tree mortality that they saw, and you can see that they found that in the very beginning you have most of the trees remaining very healthy, even 5 or 6 years into an infestation, and then it starts to go up very rapidly. And this is typical for what we'll see in infestations where the trees look very healthy and then they all start to die at once as the EAB start to overwhelm those trees. Finally, I touched upon this when talking about some of the other symptoms of EAB infestations, but other pests and diseases can cause symptoms similar to EAB. Here's a tree with ash yellows, which is showing some crown dieback, some thinning leaves, another tree with ash yellows, which is showing epicormic shoots. So this is why we say when you're looking for new infestations in an area where EAB has not been confirmed before, to always confirm EAB, because you may not be sure without that confirmation that what you're releasing on is actually EAB, and you need to release parasitoids in EAB infested trees or they won't establish. So things like water table changes, other native beetle species, ash yellows, diseases like that can also cause signs that look similar to an EAB infestation, which is why we stress when possible peeling back the bark and confirming the larvae themselves so that you know what you're going after is EAB. Okay, so how do you confirm that you have EAB at your site? Felling and peeling a tree is going to be your best option to look for EAB, although that is not always feasible for cutting down trees. It can be dangerous, and it requires people with sawyer skills. So you can also just peel the base of trees that have signs of damage if you don't want to cut down a tree. And another option, which works well for street trees, but it can also work for forested trees as well, it's just that sometimes their branches are higher up, is that you can cut and peel individual branches to look for signs of EAB. So you can either use a ladder and a saw or a pole saw and cut branches and peel those instead of felling an entire tree. Trapping can give you an indication if you have EAB nearby. If you have a number of sites that you want to know if there is EAB coming into the area, you could use trapping as a method of knowing that yes, EAB is flying nearby your sites, but peeling is going to be the only way that you can confirm that it's EAB. Peeling is the only way to confirm that it's actually EAB infesting the trees within your site. Trapping just tells you that the beetles are flying in the area, and a lot of the signs of damage that we talk about show you that the tree is sick, but until you can peel and confirm those EAB larvae inside the tree, you could have trees dying for other reasons or beetles flying nearby without an active infestation within your site. So to go along with that, we're now going to go through how do you identify EAB larvae and compare it to some of the native beetle larvae you may come across while peeling trees. So EAB larvae are in the jewel beetle family. Jewel beetle larvae in general have these flat cross sections. They look more like a pancake compared to cerambycid larvae, which we'll talk about later, and some other beetle larvae that have very round cross sections and are round, thick grubs. EAB larvae and jewel beetle larvae in general are very flat. With EAB, the color is this like creamy white color, and they have 10 bell-shaped segments. The segments in EAB are unique for this beetle species, that they are what we say bell-shaped. At the top, they're smaller and they flare out at the base of each segment. And so that is distinct for the EAB larva. Jewel beetles in general, and a good characteristic when looking to confirm EAB, is that they have a small pair of structures at the end called the urogymphi, and a lot of the other native cerambycids, other beetles, won't have this structure at the end. So when you're looking for EAB larvae, you're looking for those flat larvae with the bell-shaped segments, the structure at the end, and typically S-shaped galleries. So the S-shaped galleries are very typical for how an EAB gallery will form. So there may come a time when you need to know the stages of the larva that you're looking at. So I thought I would go over those now. This picture here is from Camaro et al. 2012. It's a great resource if you want more photos and more details about the different instars. And if you can't access it online, it is available through the USDA repository. Please contact me and I'd be happy to send it along. So I find it most helpful to look at both body size and head shape when determining instars. First instars are very small, They're on around 1/4 of an inch or 6 millimeters, and they have a very small head with very small mouth parts. Second instars, the mouth parts start to be more exposed. The head shape takes on more of an angle, and they're about 10 millimeters or 1/2 an inch in size. Third instars start to reach about an inch in size. The range is 27 to 35 millimeters. And their mouth parts, again, are larger. The head is, in my opinion, more round than in the second instar. And the cheeks extend a little bit beyond the head, but not very much, compared to the fourth instar, where the cheeks extend much farther around the head. The mouth parts themselves are also more exposed, but the head shape, to me, is very distinctly different in the fourth instar versus the third instar, which helps tell them apart. Fourth instars also tend to be bigger. 30 to 36 millimeters is what they discuss in Camaro et al., but you can see that there's a range of overlap between a particularly large third and a small fourth. So head shape, I think, in this case, is a better indicator between the third and the fourth instar. Finally, J larvae are fourth instars that have gone into their overwintering chambers, so you can easily distinguish them by their placement within the tree. So if the larva is not directly under the bark, but is instead starting to go into the wood, and it has started to bend itself over like this, which gives it that characteristic J shape, then we would call this a J larva. If it starts to condense down, we call it a pupa. But if it just remains as a larva bent over, then it's a J larva. So that's how you can distinguish the J instar. Here are some more useful photos showing those different stages that we talked about before. And here you can see that some of the larvae show variable lengths depending on how quickly they're feeding and growing. But again, the head shape tends to change and become wider as they get older. So these third instars have those smaller faces, and then the fourth instars tend to have those wider cheeks compared to their red mouth parts. Here are some other photos showing those J larvae, which are fourth instars in overwintering chambers. Another photo showing a J larva, and those typical S-shaped galleries that we talked about before. So now I'm going to talk about some of the EAB lookalikes that you may also see when peeling sick trees and trying to diagnose a new infestation. So the red-headed ash borer or the banded ash borer larvae are two cerambicids that you might see. They are longhorn beetles in the family Neoclitus, and they attack stressed or dying ash trees. They typically have an off-white or sometimes yellowish color to them, and the larvae are more tightly segmented than EAB, and they also have a round cross-section. So they're very round larvae. They don't look flat like pancakes. Another distinct difference for these larvae is that they don't have any urogymphy, no red structure at the end. It's all white at the end. But again, one of the most common ways to identify these is that they're very round, chunky larvae. So native longhorn beetles will not have galleries that are S-shaped. Superficially at younger instars, you may see some feeding back and forth, but it's not going to have a typical S-shape like EAB does. And if you look at enough of the galleries, they're going to be much more irregular than an EAB gallery. Additionally, the later instars of native longhorn beetles will feed deep into the wood. Here's a cross-section showing that deep feeding in galleries that go all the way into the center of the tree. And what you'll see typically on the outside is these really big holes that show a larva has gone deeper into the wood. And you won't see that with EAB. Their overwintering chambers, they pack up with frass. Cerambicids, you'll see these deep holes. Additionally, cerambicids have round exit holes, and compared to EAB, which has those D-shaped exit holes. If you see rounder, larger exit holes, that's more typical of native longhorn beetles. Another insect that you might see is some of the native clearwing moths. So we have the banded ash clearwing and the ash lilac borer. These are both moths in the clearwing family, so they can attack healthy ash trees. The galleries for these will extend deep into the wood. They also have very round larvae, and they expel frass from the tree, which can accumulate in bark crevices, bark crotches, and on the ground. But if you see an insect that's pushing frass out of the tree as it's feeding, it would be something like a clearwing moth. EAB never pushes the frass out of the tree. It just packs those galleries that you see with the frass. So here you can see a very different head shape on the ash borer larva and a very round body, tightly segmented, no bell shape. Another EAB lookalike you might see is a carpenter worm. So we have the carpenter worm moth or the locust borer, which are both moths in the Coccidae family. They prefer stressed trees, although an infestation from a carpenter worm rarely kills a tree. The galleries for these worms extends deep into the wood, which again is uncharacteristic of emerald ash borer. And the larvae themselves are very round. They have these large head capsules and very round larvae. And their holes, their feeding tunnels, are open, and they also expel frass from the tree, unlike EAB. So here you can see some frass pushing out of this tree. And they have very large exit holes, almost the size of a quarter. There is one native Agrylus that you may see within an ash tree, Agrylus subcinctus. Since it's a jewel beetle, it does look similar to an EAB, except it's significantly smaller. So here shows the relative sizes. The largest instar for a Agrylus subcinctus is still smaller than a second instar EAB. In addition, the eggs in the immature stages are only found on recently dead ash twigs, so you would only see them if you're peeling branches, not if you're peeling tree trunks. If you did see it though, it doesn't have the bell shapes. It has just really straight segments with no size difference between the start and the end. And again, it's significantly smaller than emerald ash borer larvae. And now I'm going to talk about the history of biological control of the emerald ash borer. So the initial control strategies for EAB focused on mechanical removal, regulatory control, and also some chemical control. This approach has been used with other insects like the Asian longhorn beetle, which we have been able to successfully eradicate from certain cities. Unfortunately, due to EAB's life cycle and how quickly it flies and how many offspring it produces, this strategy, despite really great efforts, was not successful and EAB continued to spread. So what is classical biological control? This is the importation and release of specialized natural enemies from a pest's native range. It has been successful in controlling several invasive species, including winter moth, ash whitefly, eucalyptus longhorn borer, purple loosestrife, and clamato weed, and it can offer sustained, cost-effective, and targeted control. So why use biological control? Biological control is used when you have a species that's not native, it's been established for at least 5 years, and is causing significant economic or ecological damage, where eradication is deemed not possible and control through other means is not possible. So what are the steps that are involved in classic biological control? The first is to study the biology of the invasive pest, and we can do this in both its native area and its introduced area because the biology may differ. 2 is to start surveying for native natural enemies in that introduced range. So in this case, seeing if there's anything already in the United States controlling emerald ash borer. 3 is to do foreign exploration for natural enemies in the pest's native range. In this case, that would be China and Russia, places where emerald ash borer is naturally found. 4 is to select potential biological control agents. And then 5 is to import and study those biocontrol agents in a quarantine facility in a controlled lab environment. And this is where you do host specificity testing to look to see if you have a biocontrol agent that is specific to the pest in question and won't attack native species. If you find a biocontrol that is host specific, then you can prepare a release justification document, which has to be approved by higher ups. 7 is to request and receive permits for field release. And then 8 is to select diverse field sites for your releases to start mass rearing the biocontrol agents and to start releasing them. And this is the stage that we're at for emerald ash borer. And then 9 is that we also, even after releases have started, continue to evaluate the efficacy and the control that those biocontrol agents are having, and also study just to make sure we don't find any non-target effects from those biocontrol agents that we release. So we continue to do research after the fact, and that can help inform future research. So going back to that second step in a classical biological control release, we did look for natural enemies of EAB that were already in the US in Michigan in 2003 and 2004. And although there were some parasitoids found, altogether they were having less than 1% parasitism of all of the natural enemies on the EAB population, and it was deemed that they were not having a significant controlling effect. In 2003, exploration for natural enemies of EAB in its native range were also initiated, and the exploration occurred in multiple countries where EAB is native: Mongolia, Russia, Japan, South Korea, and China all had research done to look at natural enemies there. My boss, Dr. Julie Gould, focused her research in China, so I have some maps provided by her showing the provinces that were explored from 2003 to 2007. This shows some of the collaborators that they worked with and the different regions that were looked at. In this map, you can see where some of the biocontrol agents that we use today were initially found. So Tetrasticus and Oobius were found in these regions, and Spatheus agrilli was found in a further south region. Sclerodermis puparia is another native control that was found in China. However, it was never considered as a biocontrol agent because a large percentage of the females are wingless, so it would be a poor dispersing agent, which wouldn't be suitable for biocontrol. In addition, the parasitism that they found even in the native range was very low, which would not be great for a biocontrol agent. And members of this family have been known to sting humans, so there was some human impacts as well that they were concerned about. This is just to say that when they're looking at biocontrol agents, before they bring them into testing, they're looking at how the natural history is of those species to see if they would be suitable biocontrols before they start the testing. So there's a lot of research that goes into the species when they're discovering them in their native range. This slide here shows all the parasitoids that were considered and eventually approved for biocontrol of EAB. So Tetrasticus planopenense and Spatheus agrilli were both approved in 2007. Spatheus galeni, which is from the Russian Far East, was discovered later, tested later, but was approved in 2015. All of these are gregarious larval parasitoids, so they have multiple offspring per EAB larva, and they attack the EAB larval stage. Oobius agrilli was also approved in 2007, and it's a solitary egg parasitoid that goes after the EAB eggs. One of the key stages in approving a biocontrol agent is testing that host specificity to make sure that it will attack the pest in question but will not attack a native species. So I wanted to go through some of the summary of evidence for the different EAB parasitoids. Tetrasticus planopenense did not attack any of the non-target species that was presented in its no-choice test, and testing in China showed that it was never reared out of any other Agrelli jewel beetle species. Spathias galeni only attacked EAB and gold-spotted oak borer, which is another invasive borer in the US. And even then, it only attacked it at significantly lower rates than what it was found to attack EAB at. Spathias agrelli was found to attack significantly more EAB than any non-target insects in its laboratory no-choice and choice testing, and follow-up testing was done with an olfactometer, which tests what the species is attracted to for smells, and Spathias agrelli was only attracted to ash and willow. It wasn't attracted to other tree species. Additionally, testing in China showed that it was never reared from any other Agrelli jewel beetle species. Obius agrelli was found to significantly prefer the eggs of the EAB on ash logs over any of those of native jewel beetle species. Next, I want to talk about the EAB parasitoid life cycles for each of the parasitoids that we work with. So Obius agrelli is an egg parasitoid, meaning it goes after the egg stage of EAB. It is parthenogenic, which means it can reproduce asexually, and in the US, only females are released which produce other females. This species parasitized up to 60% of the eggs in China, making us believe it would be a good control agent for use in the US. Obius has at least 2 generations during the EAB egg-laying season, and each Obius adult can parasitize up to 80 EAB eggs during its lifetime. The Obius will spend their winter as larvae inside EAB eggs and emerge as adults the following spring. Tetrasticus planopenense is a larval parasitoid, which means that it attacks the larval stage of EAB. It's a gregarious parasitoid, which means it produces multiple offspring per host, and it's internal. So it lays its eggs inside of an EAB larva, and the Tetrasticus larva will develop for a time inside of the EAB larva. The EAB will actually continue to feed for a while after the Tetrasticus has laid its eggs, and eventually the larva will develop to the point where it kills the EAB larva. And then as they continue to develop, eventually they will burst forth from the EAB larva, kind of like in the movie Alien, and then finish their development outside of the EAB, inside, but still inside that EAB gallery. Now, compared to Spathias, which we'll talk about next, Tetrasticus does not develop cocoons. They will just pupate inside the gallery with no cocoons, and then emerge from the gallery as adults. One EAB larva can produce up to 130 adult Tetrasticus, so they have a really high reproductive value. Due to their short ovipositor size, they're most successful at attacking trees that are under 6 inches in diameter at breast height, so trees with thinner bark, or the tree limbs of larger trees. Spathias agrelli is a larval parasitoid as well. It is gregarious, again, which means it produces multiple offspring per host, but it's an external parasitoid. So both of the Spathias species actually will paralyze the EAB when they lay their eggs. They lay their eggs on the outside of the larva, and the Spathias larvae feed on the outside of the EAB, and then they feed inwards. So agrelli was found to be parasitizing up to 90% of the EAB larvae in Beijing, China, which made us think that it could be a suitable biocontrol agent for here. It's only being released in experimental plots in the south because it's proven very difficult to establish. And I will show you here, just like with galeni, once it continues its feeding and goes to pupate, it creates these silky cocoons, which are a diagnostic feature for Spathias species. So even after the species emerge, they typically leave behind these cocoons. Spathias galeni is another Spathius species. It's also a larval parasitoid that's gregarious and an external parasitoid. The difference with Galeni is that it's from the Russian Far East, and that climate is a more similar climate match to the northern US, and it does not diapause, which is in contrast to Spathius agrilli, so it comes out early in the spring. This means that we release it in areas where the EAB exhibits a 2-year life cycle, where you have EAB larvae present in the spring, because once it emerges, it needs those larvae to be able to develop in and continue its life cycle. So it's released in those areas that have larvae in the spring, like Trichasticus is. So next I'm going to be talking about how to identify ash trees. So the genus for ash is also known as Fraxinus, and it's spread across Europe, Asia, and North America. All North American Fraxinus species to date have been susceptible to EAB, including white ash, green ash, black ash, blue ash, and pumpkin ash. The genus is typically dioecious, which means it has separate female and male trees, and it's typically a wind-pollinated genus. So here you can see a female flower versus a male flower for one of the species. So how to identify ash by the buds? The different species have slightly different characteristics, but what you'd be looking for in an ash is that they have these brown terminal buds that kind of look like chocolate chips, and then they have these U or D-shaped leaf scars with a single band, as opposed to hickory, which has different looking leaf scars, but in the ash it's just a single band. So here you can see white ash, black ash, and green ash showing pretty similar buds. There are slight differences if you want to get into identifying by species, but for the most part, for our purposes, we're just trying to identify ash in general. So you can also identify ash by its leaves. Again, the different species have slightly different leaf characteristics, but all ash trees will show leaves that are opposite and pinnately compound. So the leaves will vary in size and in the number of leaflets depending on the species, from 7 to 13. So black ash tends to have more leaflets than white ash or green ash, but they'll all have these opposite leaves. So ash can also be identified very easily by their seeds. They have these single samaras as opposed to maples, which have those double samaras. And then again, if you need to get into different species, there are characteristics that distinguish the different species, but we're not going to get into that here. You would just be looking for these single samaras that are often kind of bunched together on the tree. Here's a photo showing some of the differences between the ash seeds, but again, they all have that kind of typical shape. Blue ash, there's a resource here if you need to get into it more. It's actually kind of very interesting looking because it has these boxed twigs. And then Carolina ash is a species you may find down south, which has much wider samaras than any of the other species. And if you need to know more about it, there are some resources here as well. So its terminal buds look a little bit different, not quite as chocolate chippy, but I still think they're pretty typical of what you would see in Fraxinus. But there are some more resources if you're trying to identify Carolina ash in your area. So ash, identified by bark, has across the species a typically furrowed bark look. It can be said to be diamond patterned in white ash. It's pretty distinctly diamond patterned. Green ash has kind of less prominent furrows, and then black ash can be kind of quirky and scaly looking. Additionally, younger ash can look more smooth This is a young white ash, which is completely smooth. A young green ash still has some furrows, but a lot less than a mature tree. Black ash can also sometimes have a smooth appearance, even in mature trees, at least in sections of the tree. And here are some more photos just of black ash showing how variable its bark can be. This is from a site up in upstate New York, where we're doing a study where we needed to properly identify and tell apart black ash versus green ash. For purposes of releasing biocontrol agents, it doesn't matter what species of ash you're releasing on. So the goal here is not to have you confidently say with 100% accuracy what species of ash you're using, just to know that you are using ash. But that said, if you do need to find black ash in particular, we've spent a lot of time developing a guide for that. So please reach out and contact us if you need more photos of black ash. I just wanted to show you that in general, the bark can be variable in species, particularly in black ash. So there are some ash lookalikes by bark that might be mistaken for ash when you're looking at the texture. So oak bark can also kind of look furrowed and a little diamondy. Norway maple, in particular, the maples can also have more of a furrowed look like white ash can, and then elm can also have this kind of furrowed pattern as well. If you push an elm, the bark is a lot softer than most ash species, except for black ash, which does also have soft bark. So I would say the most common species that gets confused for an ash, and I've seen it tagged and bolts released on it, is a hickory. So the leaves are very similar, and often you're looking at the leaves, they're very far away, so you have to look really closely to see whether or not those leaflets are exactly opposite or slightly alternate. But with hickories, those leaflets are going to be slightly alternate. They're not going to be exactly opposite from each other like they are in ash. Also, hickory tends to have wider leaflets near the top. Ash, the leaflets are generally narrower and of similar size on the leaf, versus hickory, the top one tends to be much larger in a lot of the species. Not always, but in a lot of the species. Here are some photos of the leaves. Again, you can see similar size in the ash. And here in the hickory, you can see that the top one is a lot larger and the top three are a lot larger than the bottom leaflets. You can also look at the buds when you're trying to tell apart hickories and ash. So again, the bud scars that we talked about before have like one line in them, versus in hickory it kind of looks like a little smiley face here. You don't see that in ash. Additionally, most of the ash are some degree of brown buds, and in the hickories, well, in bitternut hickories, they have these like bright yellow thin buds that aren't short and stocky like they are in ash. In shagbark hickory, they have these kind of big white buds, and you can see that face really distinctively here. On the shagbark hickory, again, compared to the ash, there's no face. It's just a single line. So another species that may look similar to ash when the leaves are present on the tree is box elder. This is also sometimes known as the ash leaf maple because it has compound leaves as well, and they're also opposite. Now, some of the leaves may have up to 5 leaflets, but they'll never have more than that. In addition, the way that you can tell a box elder is that if you look at enough of the leaves, some of them will have mittens, and you never see that in ash. You never see the toothy mitten shape. That is a box elder. Additionally, if you see seeds on it, the box elders have those paired samaras that you find in maples. versus the single samaras and clusters that you see in ash. So here you can see the ash and the box elder branches without leaves on them. Again, they both have opposite branching, so they can be hard to tell apart. I tend to think that ash has a little bit more of a curve to it than the box elders do, but that's kind of a hard characteristic to go by. I think the easiest way to tell them apart in the winter is if you can look at the buds. So ash buds, again, have that dark chocolate brown shape versus box elder. They have smooth twigs that are generally green to greenish purple, and they have these whitish buds that are much lighter than the bark, and they just look different than ash buds. Here are some branches showing other maple species, which again also have opposite branching, so it can look artificially similar to ash. The buds for other maple species are distinctly scaled. You can see scales on them. You don't see that with ash buds. And then some of the species like red maple will have these very bright red buds or these greenish red buds, and you won't see that in ash either. Here's another picture showing just the buds. So again, chocolate chip buds versus red or dark red maroony buds of some of the other maple species. Another lookalike people have told me is walnut. I will say black ash is the most easy to look like walnut because black ash has the most leaflets. But walnuts in general will have 9 to 21 leaflets, which is much more than ash typically have, except like I said, black ash, which can have up to, I think, 11 or 13 leaflets. Again, walnut is going to have alternate branching, unlike ash's opposite branching. It's also going to have this very distinctly chambered pith, which you would not see in ash. And people have told me that walnut has this spicy odor, which you can kind of detect by crushing a leaf and smelling it, that you will not see in ash either. So we'll circle back to oaks here. I know I talked about it when I said that the bark is similar, but you know, with oaks and elms, they have alternate branching. But a lot of times the alternate branching for these trees where all the branches are up in the canopy can be really hard to tell without closer inspection and or binoculars. Here you can see at the edge, these two branches on an oak are alternate, but they're very close, so from far away it would be easy to see them as opposite. So this is just to say that it's a lot easier to sit here with nice pictures and say, oh yes, alternate, opposite, but in the field it's just going to maybe take a little bit more practice and a little closer inspection and binoculars to feel comfortable identifying. And again, if you have the opportunity to go out with leaves on your trees, it will be a lot easier for some of these lookalikes that look very different when there's leaves on the trees. And then here's a photo showing clustered buds on a couple of oak species. Many of the oak species have clustered buds at their end, which is in contrast to ash, which has a single terminal bud. Okay, now module 2 is where we'll get really in depth into participation in the program and what requirements we're looking for in sites where releases can occur. But because it's such a big question, I just wanted to start with a brief overview here at the end of module 1 with what we're looking for participation in a release program. All right, so for participation in the release program, you know, for our participants, we have government agencies, all levels, universities, even organizations with interest in forest health have participated. And the time requirement is generally 2 years of seasonal parasitoid releases and then 2 years of recovery work for those parasitoids. It sometimes has been pushed into 3 years of releases or recovery. especially during the pandemic, some things had to get staggered, but we try and keep it as 2 years release as 2 years recovery. Release sites can be public or collective property. We try to avoid personal private property, and I'll talk more about why later, although private property has been used on some occasions. And then, of course, you can find out all these requirements and more in our guidelines. Now we'll be taking a 15-minute break before our next and last section before the live quiz at the end. Our last section will be on the impacts of biocontrol agents, and it will be led by Dr. Julie Gould. So next up is going to be talking my wonderful boss, Dr. Julie Gould. She'll be talking to you about evaluating the impacts of EAB biocontrol. So I wanted to just give her a short introduction here. Dr. Julie Gould has been working on biocontrol of insect pests since her doctorate research on gypsy moth at the University of Massachusetts in the 1980s. As a postdoc, she was a member of the team that successfully controlled the ash whitefly using biocontrol. And she's also worked on the biocontrol of other insect pests, including Russian wheat aphid, Eucalyptus longhorn borer, and the weedy saltcedar plant. In 2001, she moved to the APHIS Science and Technology Laboratory in Massachusetts, where she currently is. And she is a lead scientist now, the lead on emerald ash borer, biological control, Asian longhorn beetle, and Polyphicus shot-whore borer biocontrol. And she's a co-lead on Rosso cane scale and spotted lanternfly biological control. So she's involved with a lot of programs. She plays an active role in all aspects of these biocontrol programs, including the foreign exploration for native natural enemies, host specificity testing, development of rearing technologies, release, and post-release evaluation.
Us for this training on EAB biocontrol. We really appreciate all the support that everybody provides, both in releasing the parasitoids and doing the follow-up work to find out if they're establishing and doing any good. It's very important, since we're spending taxpayer dollars, that we evaluate the impact of the program and make sure that it's doing good for the public. So there are several steps that we have taken in evaluating the success of the EAB biocontrol project. The first step is to look at reproduction. Are the insects reproducing in the field? That's step number one. But even just because they're reproducing doesn't mean they're having an effect. We need to make sure that they're establishing, that they're lasting in the environment for year after year. We usually define establishment as recovery at a particular site at least 2 years after the last release. For many of these parasitoids, that's up to 8 generations. Also, are they dispersing? Are they moving out on their own? We can't release everywhere. The parasitoids need to get around on their own. Number 4, are they reducing the density of EAB? That, of course, is important, with the ultimate goal of helping ash trees survive. And finally, we need to integrate biocontrol with other management techniques so that we can have even more bang for our buck. And these are the topics I'm going to be discussing in the rest of my talk. So in order to sample for reproduction and establishment, we have developed several methods, all of which work and are suited to different needs and to different circumstances. We can put either logs or bark into tubes, and the parasitoid adults, once they emerge, will get caught in the little cups. We can scrape the bark off of trees and then dry it and sift it, and the EAB eggs will eventually be in the duff that we sift out. It's very time consuming, but it does tell us levels of parasitism, which is something that other methods don't give us. Yellow pan traps have been a standard technique for looking for parasitoid establishment and recovery for many years. They're very cheap, they're easy to use. I personally love going through the samples looking for the parasitoids because you collect so many interesting insects. But they don't tell you a percentage parasitism. They just tell you whether the insects are persisting at a site. We have bark sifting for Oobius agrilli, the egg parasitoid, and we have tree peeling for larval parasitoids. You can see that there are galleries exposed on this tree, and we are able to tell from the larvae of the EAB whether or not they're parasitized. So we have been doing, we meaning all of you, have been doing these sampling techniques throughout the country since we began releases in 2007. We've found Oobius agrilli in 44 counties in 15 states. We recovered, with the emphasis on recovered, Spathius agrilli in 22 counties in 9 states. Spathius galeni has just started being released pretty recently, but we've found it in 12 counties in 8 states. Tetrasticus is 70 counties in 18 states. And you can see that we've recovered at least one of the parasitoid species in 20 states. But as I mentioned, recovery does not equal establishment, and Spathius agrilli, to our knowledge, has never established yet. We are still working on it and still looking. We just haven't found it yet. So climate models show that in Tianjin, China, where we collected Spathius agrilli, it's more suitable or similar to the central and southeastern part of the United States. So the EAB program decided to save resources and release Spathius agrilli only south of the 40th parallel. The problem, of course, though, is that Tetrasticus planopenisi has a very short ovipositor and is restricted to small diameter ash trunks and branches. So we needed something large. Fortunately, we found another species in the genus Spathius, Spathius galeni. It also has a very long ovipositor that's necessary to attack EAB through thick bark. And it came from Vladivostok, Russia, which has a climate that's much more similar to the northern US than is the climate where Spathius agrilli was collected. It's also quite species specific. We found that out in our testing. And we submitted a release petition which got unanimous approval of the North American Plant Protection Organization panel, and releases of this species began in 2015. It's been recovered now in 9 states throughout the country. One of the things I hinted at earlier was the fact that Spathius agrilli was not establishing anywhere in the north, and that there are climate matching considerations that need to be taken into consideration. And we have studied the phenology of EAB and its parasitoids in the south, in Tennessee, and in the north in New York. And we found that in Tennessee, EAB exhibits a 1-year life cycle. The majority of the emerald ash borer larvae are overwintering as J larvae. These larvae are very deep in the bark, and they are not available to parasitoids. And these parasitoids emerge early in the spring. And so, but in New York, EAB populations overwinter in all larval stages. So in New York in the spring, you've got plenty of larvae for the early emerging parasitoids to attack. So the early emerging parasitoids are Spathius galeni and Tetrasticus planopenisi, and they emerge in the spring where hosts are only available in the north, and they're unlikely to establish where EAB has a 1-year life cycle. Spathius agrilli, on the other hand, emerges a month after EAB, and it's well synchronized with EAB in the south, where there's a 1-year life cycle of EAB. In the north, our studies found that it has a dead-end generation in the fall, and that is probably what is leading to its lack of establishment. So we expect Spathius galeni and Tetrasticus to establish in the north and Spathius agrilli to establish. As I mentioned, the different species have different environmental requirements. Spathius galeni and Tetrasticus establish better in the north. We also do know that Oobius agrilli has specific diapause requirements, and it also has a gradient over which we'd expect it to establish better, but we're still working on learning what that is. So we've got a handle on where we should be releasing these parasitoids and where they're likely to establish, but we wanted to know, do they move? Do they disperse well? We did a study out near Rochester, New York, where we followed emerald ash borer and Tetrasticus planopenisi along a greenway. This map shows a greenway, and it has ash all along it. It's actually an old canal where they used to pull boats with mules. And you can see that the emerald ash borers caught in traps in number A, which is the first year of the study, the emerald ash borer was confined to about the northern third of this greenway, and we released parasitoids at sites R1, R2, and R3, and you can see that R3 was really where there weren't EAB yet. Then the following year, you can see where we also recovered Tetrasticus planopenisi. That's where the stars are shown. And you can also see that there was a rating of EAB mortality of trees, with red being severe and green being minor. And you can see that Tetrasticus is limited to the upper part of the greenway, where there are emerald ash borer populations. You can even see that in the furthest south star where Tetrasticus was found, you do have more emerald ash borer at that little spot. And you can see that we follow year after year. The emerald ash borer is moving south along this gradient, and the Tetrasticus are following right along behind it. It was really a nice demonstration that Tetrasticus is really tracking and keeping up with its host as it spreads. Alobius agrilli also spreads, and we've done studies where we had release and control plots, but we found that it spreads more slowly. And it's been found to spread over 1 kilometer, but it takes it a few years to go that far. So another study that looked at the dispersal of the larval parasitoids was done in Connecticut and New York using the technique of sentinel logs, which is something Theresa is going to talk about later, which is we put larvae in these logs and we keep the logs wet. At the bottom there's a cup containing rock wool, and there's EAB larvae in these sticks, and we put them out and the parasitoids find them really, really well. And so we did 2 studies where we had a release site, and then there were 2 transects that went out from the release point, and we put sentinel logs along that transect, and the transects were 14 kilometers long. And what we found was that both Tetrasticus planopenisi and Spathius galeni were detected throughout most of the transects. Spathius galeni was found up to 10 kilometers away, and Tetrasticus planopenisi was detected up to 14 kilometers away in both transects in New York. And similarly in Connecticut, both species were detected 14 kilometers away from the release site. And what we found was in 2021, a graduate student I'm working with had set up some yellow pan traps 8 kilometers away from this release site on the other side of the Hudson River. So this was on the east side. The yellow pan traps were set up 8 kilometers away on the western side of the Hudson River, and there was Spathius galeni. It had moved across the very large Hudson River. So we do know that parasitoids will establish, they will move. Will they do anything to slow EAB population growth? One of the techniques we use is called life table analysis, where we look at the mortality factors that affect EAB and ask how the parasitoids rank in terms of mortality factors. So Jian Duan with ARS has been doing some long-term studies in Michigan, and he found that at these sites between 2008, remember this was when the parasitoids were first approved for release in 2007, so he started this study in 2008, and he found that the EAB population peaked at both release and control plots in 2009 and declined ever since. And by 2014, EAB densities were quite low. They were below 10 per meter square. But as the EAB density declined, parasitism increased. This is parasitism by Tetrasticus planopenisi, which by 2014 was approaching 30%. And again, in both release and control plots, as we saw before, they move very, very rapidly. So we also found that woodpecker predation was fairly steady. Even as EAB densities declined, woodpecker predation stayed pretty steady at about 40% of the EAB were killed each year. So the problem, though, is that even though the density of EAB declined and parasitism increased, the mature ash trees in our release sites died. That's because we're releasing thousands of parasitoids, but there are millions of EAB out there. And really the future of ash as a viable component of North American forests depends on the survival of ash saplings following the initial outbreak. A forest like this with all the big dead trees will have lots of ash saplings, and they're too small initially for EAB to attack them. So we have been focusing on trying to see if the emerald ash borer parasitoids, once they established, if they can save these smaller trees. So I did a study in New York looking at the impact of larval parasitoids in woodpeckers and white ash saplings, and I'd like to say that the results that I found were almost identical to what they found in green ash forests in Michigan. So 5 years after the parasitoids were released, the density of EAB in ash saplings was between 0 and 4 larvae per meter squared, so it was quite low. And over 70% of the ash saplings had no fresh EAB galleries at all. They even had never had an EAB in them, or they had had an old gallery that had calloused over, and they had no fresh galleries. The mortality of 4th instar EAB was 80 to 100%. And it was mostly from Tetrasticus planopenisi and woodpeckers. In eastern New York, where the EAB infestation was a little newer, we did get survival of EAB, a little over 20% by the 4th instar. But in western New York, we had 6 sites, 20 trees per site, so 120 trees that we sampled, and we did not have a single living 4th instar. You can see from this graph, though, that it's really in the 4th instar that all the mortality happens. There's not a lot of mortality of 1st, 2nd, and 3rd instar EAB. All of this mortality is happening from woodpeckers and parasitoids in the 4th instar. So if you look at the net reproductive rate of EAB with and without these natural enemies, a net reproductive rate, which is R0, the term R0 here, if it equals 1, it equals a stable population. Over 1, the population is growing. Under 1, it's declining. Well, we sampled in the spring of 2017, and what we actually found was the population in eastern New York, assuming no other mortality factors. We're not accounting for oobius parasitism. We're not accounting for adult death. Only looking at mortality of larvae, the population would be growing at 4 times. In western New York, that was 0. As I said, we saw no growth whatsoever. However, even in eastern New York, without the parasitoids, the population increase would be 7 times. Without woodpeckers, it would have been 13 times, and without both, the population would have been growing 16 times. So you can see that even in eastern New York, there was quite a reduction in the population growth of emerald ash borer, and in western New York, it would have been almost 8 times, whereas it was 0. There was no reproduction. Another thing that we found during our study was that the larger mature trees were dying at a much faster rate than were the saplings in both the release and control plots. The big trees, between 40 and 60% of them were dead during the 5 years of the study, whereas fewer than 30% of the saplings died during the same period of time. Dying. And it was really irrespective of whether we called it a mature tree or a sapling. The bigger the tree, the more likely it was to die. But the big trees were dying from EAB. 90% of the big trees that died had signs of EAB. Whereas saplings die of other things. Not all saplings become big trees. And in fact, a lot of the saplings were not dying from EAB. So they were just having natural death. So really, we can focus on trying to save this cohort of saplings, which is less susceptible to EAB, and we have a lot of mortality of EAB in saplings from the natural enemies. So in conclusion from this study, we've added to an increasing body of evidence that parasitoids are contributing to the suppression of EAB population growth in saplings. Following the parasitoid release, the EAB density declined, parasitism increased. The majority of saplings were uninfested. Most of the fourth instar larvae were killed by Tetrasticus planopenisi or woodpeckers, and the mortality of saplings was less than that of mature trees, and most died of something other than EAB. The problem, of course, is that Tetrasticus has a short ovipositor, and at the time that was the only parasitoid we had in the system. It can only parasitize through the thin bark of these small trees. So what happens when the trees get big? Because Spatheus agrilli, which does have a long ovipositor, did not establish. Well, again, we have Spatheus galeni, and it has a long ovipositor, and it's really establishing well. As shown in this graph, this again was a study by ARS. It was done in New York, Connecticut, and Massachusetts. And this shows the increase in parasitism beginning in 2015 when Spatheus galeni was released at these 5 sites. And you can see that in all of the sites, the parasitism by Spatheus galeni has been climbing, and has been approaching 80%. And keep in mind, these sites also have woodpeckers active, and they also have Tetrasticus planopenisi active. And so we really are beginning to develop a system where we have a lot of mortality of emerald ash borer, and that we're hoping we can save these small trees and get them to a larger size. Finally, I'm going to talk about the management of EAB in urban areas and how we can integrate the use of different management techniques, particularly use of systemic insecticides and biocontrol. So one of the questions that I wanted to ask was, can systemic insecticides save the large ash trees, which, as we've said, in a forest will die, but can we save them in an urban area while populations of parasitoids have time to establish, disperse, and increase? And will the suppression of this EAB populations allow the parasitoids to increase more quickly relative to their host? And can we eventually stop using these insecticides because the biocontrol agents can take over and cause sufficient mortality? So we've been conducting this study in Syracuse, New York, Naperville, Illinois, and Boulder, Colorado. We have a release and a control plot. We have 3 treatments of 50 trees each. We have trees that are getting insecticides forever, or at least until I retire. short-term insecticides, which were stopped after parasitoids established, and biocontrol only or no biocontrol. And so we released parasitoids starting in 2015 and 16, and that was Tetrasticus, Spathius agrilli, and Oobius agrilli. Spathius agrilli did not establish, as we know, and so we started releasing Spathius galeni at these sites in 2017. So here are the results. Spathius agrilli did not establish. Not a surprise. The other three did, though, in all three cities. We have recovered all three parasitoids in Colorado, Illinois, and New York. Both Tetrasticus and Spathius have increased, and they have dispersed widely. The map on the right shows the recoveries of Tetrasticus planopenisi using sentinel log method, and you can see that they were literally found at every single site. Everywhere we put a sentinel log, we found Tetrasticus. Spathius galeni wasn't quite as widespread, but they had been released much later, so they didn't have as much time to build up and disperse. And we also found, these data are also from Syracuse, just as an example, but we found the percentage of logs attacked by the parasitoids was very high. In Tetrasticus planopenisi was well over 70% in the first three deployments that we used. This was in June, July, and August. And Spathius galeni built up over the summer. But we found both of the parasitoids were attacking the EAB. They found the logs, and they also parasitized most of the EAB that we had in the logs. So they, parasitized more than one EAB in each log. So parasitism levels were really high. And so the results, we also found that both Tetrasticus and Spathius increased and dispersed widely. Parasitism by Tetrasticus was between 18 and 46%. Woodpecker predation was between 21 and 67%, and EAB density fell below 10 larvae per meter squared, which is a quite low density. We also found that we were able to protect the trees. This is an ash health assessment. This is an assessment of how healthy the trees were. A green is a canopy of 1. It's a beautiful, healthy canopy. A red is 5, and 5 is dead. 4 is almost dead. And you can see that by 2020 in Syracuse, most of the trees were either dying, really dying, or dead. So the treatment that we used really did help protect the trees. We have parasitoids building up, and in 2019 we ceased insecticide treatments in our short-term treatment trees. So we are going to continue to monitor these plots and to determine whether the EAB comes back into these trees from which we've removed treatment, or whether the parasitoids can continue to keep the EAB in check. One final study that we are just getting underway is a study on forest IPM. We found that this IPM concept worked really well in the urban areas, but ash also plays a really important ecological role, especially in riparian habitats. You can see that on the left-hand photograph, there's a riparian area that's just been hammered by EAB. Ash is economically important. We make baseball bats and handles out of it, and ash also has cultural significance and economic significance to the native peoples. So even though we can't treat every tree in a forest, we felt it might be important to keep large trees alive for seed production. Ash seeds have a really short life expectancy, and it can take an ash tree 30 to 40 years to reach maturity where it produces seeds. So we wanted to be able to keep large reproductive ash trees alive in the forest. In 2020, we established 3 study plots in green ash forests in upstate New York. We hadn't yet found EAB in black ash forests, which really is our main target. So we were putting up traps and looking for EAB in black ash forests in both New York and Wisconsin. In 2021, we did find EAB at black ash sites in New York, and we started to set up some plots and to treat the trees with insecticides, and we are continuing trapping in Wisconsin. We have not yet found any sites to work at there, but we are working on it. I'd like to thank my coworkers who've kept me sane during the last 2 years of working at home during COVID. And I'd once again like to thank all of you for making my presentation possible, because much of the data that I have presented today was gathered by people like you who are helping us with this project. Thanks again for everything.
All right, first question, is this an EAB larva? Yes, no, or not sure? No, this is a red-headed ash borer larva. It's much rounder in cross-section, and its segments are very tightly put together, and they're not bell-shaped. It has no structure at the end either. Is this tree infested with EAB? Yes, no, not sure. In this case, these are photos I got from someone out in the field. This is an example of what the gallery looked like, and this is the larva, but they cut the larva, so there's no end or head to the larva. You can only tell by the middle segments. Even with just the middle segments, we can see that they're bell-shaped and it's flattened. It's not round in cross-section. So yes, I would say this is an EAB. And then the gallery here as well has a very typical S pattern that we would see with EAB. Are these EAB larvae? Yes, no, or not sure? Yes, this is 2 EAB J larvae. Because they're J larvae, they're a little bit thicker than the first through fourth instars just on the wood would be, because they're starting to get ready to pupate. But they're still mostly flattened, but you can see the bell-shaped segments very easily, which is typical with EAB larvae. And it does have the structure at the end. So yes, these are EAB. This is a larva I found peeling ash trees in Illinois. Is it an EAB? Yes, no, or not sure? So no, this is not an EAB. Its segments are rounded. There's no bell shape to it, and there's no structure at the end. And its head is also massive compared to its body, which isn't something you see with EAB. It's likely some sort of native longhorn beetle larva. Here's another larva that's been cut off, which really is easy to happen when you're peeling trees, because those draw knives are so sharp that they can easily cut into an insect. So with just the head of the larva, no tail end, is it an EAB? Yes, no, or not sure? Okay, so here's that same photo, and it's zoomed out a little bit to show you some of the other larvae. So now that you can see other larvae that are intact, does this change your opinion about whether it's EAB? Yes, no, or not sure. So I can tell that that's an EAB even with no end because it has those very bell-shaped segments and it's flattened, and it looks typical for what we see for EAB. But in this case, there's also more larvae that we pulled out of the same tree, and those do have the structure at the end and are also bell-shaped and flat, which confirms again that it's EAB. So these are photos sent to me by one of our technicians out in New York trying to confirm if there is EAB at a site. And I got these photos. And using this, would you say it's a tree infested with EAB? Yes, no, or not sure. No, this is likely damage from a native longhorn beetle. You can see these deep holes where it's feeding deep into the wood, and you do not see that with EAB. And then overall, the feeding is very irregular and it's not S-shaped, which is more typical for some other species. But the deep feeding, and here an exit hole that's large and not D-shaped, another exit hole large and not D-shaped, are good indications that it's not EAB. So based on this photo, can you tell what tree species it is? Ash, hickory, maple, walnut. Could be ash, definitely not ash. So this is actually hickory. The leaves are much larger at the end and it's not opposite, it's alternate. Can you tell the tree species? Ash, hickory, maple, walnut, not sure. Could be an ash, definitely not an ash. So yes, this is an ash. This is a black ash, so it has a lot of leaflets compared to some other species of ash. But you can see that they're all opposite here, and it also has seeds, samaras, which also are a good indication that it's an ash because it's single samaras. It's not double like you might see in maple. Is this an ash again, or is it a hickory, a maple, walnut? Could be an ash, definitely not an ash. So this is actually a walnut. If you looked closely, you'll see that it's actually alternate and not opposite, and there's a lot of leaflets. So if you found one with too many leaflets, it would be another good indication that it's walnut and not ash. This is a tree in fall, which could make it easier with some of the color keys, but also looking for trees in fall can make it harder because a lot of the leaves and features start to fall off. But is this tree an ash, a hickory, a maple, walnut? Could be an ash, definitely not an ash. So yes, this is a white ash, and the color of the leaves can be helpful here, showing that they're yellow, which is pretty common for ash, but also sometimes red in certain species, and these are yellow and red, which is kind of cool, and you can tell that they're opposite and they have the right shape leaflets.